to those of you who brave the sub-zero temperatures to join us in person today. I was a little bit skeptical, not being from central New York originally. You were a hardy bunch. This is impressive. So thanks for your attendance. My name is Mike Gladmeyer. I'm the Associate Vice President for Facilities and Campus Planning here at Hamilton College. And it's a pleasure to have you here for today's installment of our Arboretum Speaker Series. So speaking of our series, we're very interested to learn more about how our audience gets the news of these events. So if you are joining us today by Zoom, and I understand there's about 36 to 40 of you and still climbing, so thank you for your attendance virtually. If you could please indicate in the chat box whether you heard about our speaker series via email, website, newspaper, word of mouth, whatever media source that you're getting this information. We'd love to hear from you. For those of you in the audience, I think Fran has already passed out a clipboard with a simple form to also get that feedback from you, so I appreciate it very much.
40 people signed in via Zoom.
And if you are online, if you could just please make sure that you are muted. We appreciate that very much. So once we've finished mapping out our own communications network, we're going to dive into a network that is much older and, as we're about to learn, much more selective. On a morning this cold, it's natural to want to huddle together for warmth. And if you've read popular books like Suzanne Simard's Finding the Mother Tree, you might imagine the forest floor is doing just that, a vast, harmonious wood wide web, as it were, where the trees and fungi are all one big happy family sharing resources like a cozy neighborhood potluck. However, our guest today, Dr. Tom Horton, is here to give us the mycocentric reality check. As it turns out, the underground network is a bit more complicated and perhaps a bit more exclusive. According to Dr. Horton's research, the forest floor is actually less like a community potluck and more like our dining commons at lunch. There's a few students here who can attest it's patchy, it's complex, and the different species are very particular about who they interact with. In the fungal world, if you aren't part of the right clique, you might find that your invitation to the network has been revoked. But it's a true honor to introduce the man who's going to lead us through this fascinating cliquish subterranean world. Dr. Tom Horton is an emeritus professor of mycology from the SUNY College of Environmental Science and Forestry. He earned his doctorate from UC Berkeley and has spent decades studying the ecology of these hidden networks. He is a pillar of the scientific community, serving as the longtime advisor for the Central New York Mycorrhizal Society, the scientific advisor for the Mianus River Gorge Preserve, and for the last 15 years or so, the lead for the annual mushroom fair at the Beaver Lake Nature Center. He is also the recipient of the William A. Weston Award for Excellence in Teaching from the Mycorrhizal Society of America. So, while the fungi he studies may be famous for rejecting fusions and being difficult neighbors, I can promise that Dr. Horton is an incredibly engaging guide to their world. So without further ado, please join me in giving a warm welcome to Dr. Tom Horton.
That has to be the best introduction I've ever had. Wow. I don't really need to say much more. The presentation has been given. Yeah, so I got my start. I did all of my research out in California and up in Oregon, as I'll show a little bit of some of that work today. But back in 1990, I started at San Francisco State, and I took a class from a plant ecologist, and I was really intrigued at the way he thought. And I also realized that all my backpacking and observing had paid off, because everything he was saying, I thought, yeah, no, that totally makes sense. And I realized that I was kind of a plant ecologist without any training already. So I went to his office and I said, do you have any projects? Because I hadn't selected a professor yet. And he gave me a lot of interesting, he was a disturbance ecologist, gave me a lot of interesting scenarios, how ways are impacting the salt marshes in the San Francisco Bay.
PhD from University of California, Berkeley. Retired from SUNY.
Could you turn off your microphone?
Whoops, I turned off.
Thank you. So yeah, he gave me a whole bunch of projects that he was working on or needed students to work on, and I thought, no. I thought, this isn't going to work. And then he said, I have one more. I've got a chaparral species up on Mount Tamalpais across the Golden Gate, where there are Douglas fir getting established in a particular species or genus of plants and not in other locations. And I think it has to do with mycorrhizal fungi. There's good evidence of that from research up in Oregon right now. And I said, did you say fungi? Because I had gone to school, my undergrad had been at Humboldt State, Northern California, and I learned how to find mushrooms, identify them, and then eat them. So really good ones out there. Oyster mushrooms, chanterelles, porcini. Some of us know these more than others. And he said, yeah, I think it has to do with the fungi. And I thought, I think I'm your guy. So we wound up writing a grant. It got funded through, that was a USDA grant. And by the time it got funded, I'd already done the project. Because I had to come up with the methods, and things could go pitifully slow in getting funding. So it got funded. I finished my master's, left the money for somebody else to do their work, and I went over to Berkeley. I got accepted to work in Tom Bruns' lab over in Berkeley. So I'll go over a little bit more of that in a minute, but just a little bit of the backstory. By the way, my grandfather was born in Apulia Station. Yeah, not many people know it. You cross the railroad track between Tully and Vegas. So he was born in, I think, '02, and he was a dermatologist. And so I go to his house when we visit, or go to his office, and look through the microscope at fungal spores and things like that as a little kid. So apparently a spore was planted back then. I had no idea what was coming, and neither did he. So anyway, that's a little bit of the backstory. Let's see, you can see the Finding the Mother Tree, Suzanne's book, and also this pointer won't work for, oh, okay, so does that go through the Zoom? It does not. So I shouldn't use that. I should use this.
Yeah.
Okay. 1 second while I get this so it's a little bit easier to, this one, and that. There we go. So, yeah, she comes up with a mother tree. She and I were doing our PhDs right around the same time. She was up in Canada looking at mycelial networks and how carbon flows from one tree to the other, as was Peter Wallenben in Germany. They both put out these books, and they both kind of wind up talking about how the trees are talking to each other through the mycorrhizal networks. And I say that with a degree of maybe pushing it. And I'll be talking a little bit more about that today. That's kind of where I'm headed. There have been a number of, let me just say, there's been a number of critical papers that say, wait, not so fast. And I was about to put out one of those, and I went, wait, not so fast. Let's just let others do that. I don't need to be the punk. I do, but. Okay, so here's the stand of trees that my master's professor, Tom Parker, showed me early on in my master's. This is across the Golden Gate Bridge and then up into Marin Headlands, not too far from Lucas Skywalker Ranch, which, you know, for me, I didn't know what the heck it was. But when I meet Star Wars fanatics, they're like, wow, that's awesome. There was a fire here. 2 Corsair airplanes crashed during World War II above this area, and one went into the Pacific Ocean, and the other one landed somewhere around here. And in fact, you can find pieces of engine and other things in the area from that one. It started a fire. This was back in the, I think this was back in the 50s. I think it was after the war. But it wiped out everything except the chaparral species. All of these, whether it's this one or this olive one or the one back here, are burrow formers. And they resprout after the fire goes through. And the root system stayed largely intact, and they just regrow right away. The Douglas fir, which is what these Christmas tree guys are, has to re-invade. Seed has to blow back into the area. And this is a really amazing site because it says it all with this one picture. I gave this presentation one time and a very respected ecologist came up and said, that picture said it all. And I have to agree. Well, yes, I have to agree. You draw a line right there, you have the Arctostaphylos, which is manzanita. You can buy these in the store around here, Arctostaphylos uva ursi. Not very arboreal for the Arboretum folks. It's, maybe up about that tall. They can vary a bit. And if you note, the Douglas fir only got established in the 50 years or so since that fire, wherever that shrub is. At least that's what it appears to be going on. Out here, this is Adenostoma fasciculatum, chemise. It is a little needle-like leaved shrub, also burrow former, but it forms the wrong mycorrhizal associations or associates with the wrong fungi. These back here are associating with things like amanita, chanterelles, boletes, all your typical fungi if you're into that field or hobby. But out here, these are all microscopic and it's a very different group. This is a 400 million year old mutualism back here. These are more like 50 million years. They evolved much later. But they're more interesting to us because they support trees that these support trees too. Very important trees like maple, cherry, ash, some others. But if a Doug fir seed lands out here and germinates, it's probably just going to wither and die within the first season. So my job for my master's was to find a lot of places where there were adjacent patches like this. It's a very dynamic system up there. And compare the two patches for things that would support Douglas fir to get established. And so I'm trying to rule out explanations for this pattern. Why Doug fir here? Why aren't seed was landing out here? And I looked at soil moisture, slightly more moist under the Arctostaphylos, but not tremendously so. And there are places where Douglas fir was establishing an Arctostaphylos serpentine endemic called Montana, if I'm not mistaken. And it's a really nasty dry soil, but the Doug fir didn't care if the Arctostaphylos was there. It's just a different endemic species, serpentine endemic. So soil moisture, I rule it out. Light, there's no difference. In fact, probably less light under this one because it has a bigger leaf. These are, as I said, needle-like. Soil nutrients, turns out there's a lot of nitrogen out here because this one, Ceanothus cuneatus, but a lot of other Ceanothus also, they all have nitrogen-fixing nodules with bacteria. Not legumes, not pea plants, but with a different group of bacteria called Frankia. So better if you're landing out here in terms of soil nutrients. And phosphorus is all over the place. There's no pattern there. Allelopathy, if you know, you probably have some sense of something like walnut and maple not being able to establish underneath walnut. That's allelopathy, right? Walnut is a really nasty tree for seedlings that are trying to get established nearby. No difference. They're both pretty nasty for Doug fir, both in terms of germination. They inhibit germination, or it inhibits germination. Both Arctostaphylos and the others, and root growth after germination. So no difference though. And then of course there's the mycorrhizal fungi. And call it bias or whatever, but that's what I was hoping to show. And in fact, there was far more species fruiting out here. In fact, there were species fruiting in the Arctostaphylos patches with or without Douglas fir, and none here. There was one year, it was a really big rain year. We did get some, which was a kind of a shocker, and I'm not too sure what happened there. Okay, but I finished my master's, and what I really wanted to do was find out what fungi were on the seedling roots if they grew here. And that's when I started over at Berkeley, because that's what they have the ability to do. And so the idea is not to just look at the above ground pattern of succession, but to get below ground and find out what's going on. Why is this pattern happening as a result of the mycorrhizal fungi in the roots? So just a little background. plants get their nutrients from the roots, but actually they're getting it in most systems that we're looking at from the mycorrhizal fungi. This is a radish, and it's in the plant family Brassicaceae, and they don't form mycorrhizae. They evolve later, and they evolve in a way where they don't need fungi. They really get established in brand new disturbed sites, like a parking lot, right? Or an old field, baseball field or something like that. Where do you find things like mustards and broccoli and those weedy things? They're all annual, they germinate, they flower, they put out seed, and they go away. And if another species lands in there that has a mycorrhizal association, it kicks their butt. These guys can get nutrients where there's very little competition because they have really elaborate root hairs. But put them into a spot where there's mycorrhizal fungi, and you can imagine this little one is about the same scale and size as this here, and it's just not going to compete unless it could tap into the mycelial network that this mother seedling got established with. Okay. Oh yeah, that's to show. I drew those yellow lines to kind of, one thing we look at is what's called the zone of depletion. If it's in the nitrogen-rich zone and the root goes down there, it actually will become depleted for nitrogen close to the root, very close to the root here, but a little further away from the main taproot here because of those root hairs. That's their function, is to go out and get more nitrogen or whatever, phosphorus, water, etc. Okay. So, if you look at these interactions with isotopes of various nutrients, so what do plants get from the fungus? All sorts of nutrients, nitrogen, phosphorus, potassium, calcium, etc. Everything in your fertilizer, basically. NPK is the biggie. Those are the biggies, especially nitrogen and phosphorus in our systems. And so we usually focus on those two. I looked at all of them, and it looked like the fungi were important. This shows you a little sort of bait of phosphate. And that phosphate has, instead of the stable isotope that we all know and don't worry about, it had a radioactive isotope that was made in lab. And that's why if you put an X-ray film over this, it is dark. Now it's a negative, right? So I say, wow, it really lit up. No, it's dark. But you get the point. And you can see all the root tips are also lighting up because the fungus harvested the P32 here, or the phosphate, and then delivered it throughout its body. And you can see all the hot spots where there's a lot of activity. Those are the mycorrhizal root tips. You can also see sort of the hyphal interactions. These are not roots. These are hyphae. So there's a beginning of a sense of the network. This is a root, right? It's got the mycorrhizae. It comes out this way as well, and it's right over here. That's what that is. So phosphorus is harvested and dispersed throughout the body of the fungus. It also is used by the plant, so it gets above the ground also.
Get warm. Come in.
What's her name?
Lisa.
Lisa, your microphone is on and we can hear your conversation. Don't go to the bathroom now. It's all right. Okay, more importantly for this presentation, we're talking about carbon. And that's what Suzanne Simard focused in on, is the movement of carbon between plants via the mycorrhizal network. So a similar micrograph is the last one with phosphorus. Here it's with carbon that's labeled with C14. We are C12. I won't go into what that means, other than C14 gives off radiation, and you can pick it up with an X-ray film. So you grow your seedling up. See that little curve of the root system up the top? There it is on the X-ray film. So it fixed carbon. They put a bag of CO2 that was labeled with C14, and after a day or two, they put the X-ray on top of that X-ray film, and here you see it. And now you can really see the networks. This is not root system, although there are roots in there also. But also here, this is a bait of something that the fungus was interested in, so it really put a lot of its energy into that little bait area. And you can see it really works very nicely. This is all out of David Reed's lab. His lab has just done tremendous work with the isotope and watching things move through the network, if you will. To be clear, one more time, so we're looking at putting a bag of CO2 on this plant, and then that CO2 has a label of 14 carbon instead of 12, and then you cut it off and put the X-ray film, and that's how you get that image. So it fixes the carbon into sugar, and then it sends the sugar to its body, and some of it gets into the plant. Oh, apparently I had a series of slides here to say all that. So there's your bag of CO2. There's the CO2 as sugar moving below ground, and it gets dispersed around. Okay, this is a schematic of the Simard et al. Simard's research that she did up in Oregon and Canada, British Columbia. So she had 3 different plants that she put in. They were seedlings into these macrocosms, maybe a hula hoop circle. And one of them was an angiosperm birch. I'm talking to tree people here, so I probably don't need to go into great detail here. The other was a Douglas fir, conifer, and another conifer thuya, which is cedar. Birch and Douglas fir form mycorrhizal with our favorite fungi, the boletes, the chanterelles, and all of that. They are ectomycorrhizal, and her control was the thuya because it is an arbuscular mycorrhizal plant, like herbs. And so not a lot of root interaction between the thuya and the other 2. So very beautiful experiment. And she put C13 on one side and C14 on the other. She also did other experiments where she flipped that in case one of the trees was more apt to pick up the nutrients than the other. So she made sure that that wasn't an issue. And then looked later and looked for C13 over here on the Douglas fir, which was labeled with C14, and then looked for C14 over here, and looked for both in the thuya. And it turned out there was a lot of transfer between the 2, and what she called reciprocal transfer. It goes either way, between the 2 ectomycorrhizal tree hosts. Not so much in the thuya, although some. And my best guess is what happened is the mycelial networks respired and released CO2 that was labeled, and then the thuya picked it up. And so you get a radioactive signal that way. Maybe trace amounts moved into thuya too, but not enough, not compared to what was going on with 2 ecto hosts. Okay, and that led to the Wood Wide Web, which we've already heard about. And she did, the field experiments were a little bit messy, as field ecology is. It's really hard to get a good handle on some of the data, and you have to, the polite way to say it is tweak the data with fancy statistics or manipulate the data. She had a hard time getting it in to the journal because it was sort of messy. But she also did a whole bunch of lab experiments that got rid of all the noise and focused in on one or the other aspect. And it looked pretty good that she was showing the reciprocal transfer. If she shaded Doug fir, shade cloth, 90% of the shade, 90% of the light got through. Sorry, 10% of the light got through, 90% got blocked. Then there was more carbon flow to that seedling if it had been shaded. Not very often that you're going to stand where a Douglas fir seed is establishing with 90% shade. So it was still criticized. It's like, are you sure? Are you sure that's really what's going on? But she said, well, some carbon's getting over there into that little shaded seedling. Okay, that was her work. That was published in '97. That was the year I got my PhD. My work was published a couple years later, but we knew each other. You know, it's a small world. I wound up working as a postdoc in the lab that she worked in for her PhD, so we were always seeing each other and comparing notes. And I was confident that what I was doing was important because I was looking at fungi, actually mapping out the fungi below ground with genetic types. And what she was doing was important. She was looking at the material flow through those networks without paying attention to the actual structure of the network, other than you've got a host that is associated with the fungi. Let me build this idea up a little bit with the network. This is one plant, a pine, and one fungus, a psoas. Okay? Not very complicated. You can see all the hyphae, mycelial fans out here picking up nutrients in the soil. And then the root tips are bright for the seedling. So the hyphae colonize those root tips, and I'll give you a better image of that in a minute. And that's all colonized by this one species of fungus, Swirlus. Yeah, I forget what species it is. It's okay. Doesn't matter. It's all one. Here's a more complex version. And now we have 3 individual seedlings, but still one individual fungus. So it's not that complicated still, especially below ground. But note what happens. This is the mother seedling, if you will. She germinated and inoculated the soil down here with that Swirlus again. And she basically made sugar and fed all these hyphae. And they grew out to get nutrients. Then they put in this little seed. It germinated, and it got colonized by the fungus. So now we have a nice network connecting 2 trees or seedlings. And then this one germinated later, so we still have a little bit of the seed capsule there, or whatever that's called. Sorry to the arborists. And it is just getting colonized now. So let's put a bag of CO2 over the mother tree, if you will. She fixes it, makes sugar, sends that sugar to the rest of her body, about 50% below ground, 50% above ground. So she's feeding her own roots. On those roots are these mycorrhizal fungi, and they're getting fed also with that labeled CO2. Now, some of that goes over to those seedlings. They have mycorrhizal. All of this I totally agree with, but the next part is where I kind of get a little bit worried about how we're interpreting things. She would suggest that that carbon, that sugar, transfers from the fungus back to the plant. And the problem I have with that is that fungus can't make its own sugar. It has not evolved as an autotroph, right? So it can make sugar above ground, but what's the selective advantage of figuring out a way to pick up sugar from mycelium? And more importantly, from my mycocentric viewpoint, what's the selective advantage of the fungus giving up sugar, right? It should want sugar from a plant, and it should have all the mechanisms for getting that sugar from the plant root. And sometimes I give these presentations, and at this point I go into plant, membrane-bound transporters, and the fact that they're one-directional and you don't really flip them around. So if it's going fungus to plant, why would it flip and go plant to fungus? I'm sparing you all of that. They're really complicated slides, so maybe I'm sparing myself having to explain it. But we really don't know. She has never shown, or no one I know, has clearly shown what is transferred when you do see a little bit of signal up here. As I mentioned a few slides back, maybe the thuja was just getting CO2 that had been respired out, and it was fixing it. And that's why it got some of the signal. But this would suggest that it's actually sugar moving from fungus to plant root. And I don't think anybody has shown if that's true. People have shown amino acids moving across, and there could be carbon on those there is carbon on those amino acids, and maybe that's labeled, but no clear evidence that sugar is being moved across the 2 organisms in that direction. Okay, so I promised you I was going to give you a better image of the mycorrhizal interaction. One thing you could do, so let me back up. Oops. Folks have taken whole plants, I'm not going to go further, and you can see that the label shows up throughout the root system. Okay, that's good. But is it just in the hyphae, or is it also in the plant cells? Right? I don't know if I'm getting too deep here, but you can, and people have done that, And they've cut out this portion, which is all fungal. This is called the mantle. And they've cut that apart, and yeah, it's lighting up. It's got lots of the signal. I don't have an image of that here. There's also hyphae in between cells down in here. That's why they're all fat, and you can see some lines between them, septa. Okay, so let's just say it's 60% plant tissue, 40% root fungus tissue. You could manipulate the data and try to figure out if that doesn't explain it, and indeed it does in some cases. That the signal that they're picking up is all in the fungus, even at this fine scale. I forgot to build this up a little bit. This is a Lacaria on a Douglas, on a pine, and all the little white dots in this area are mycorrhizal root tips. And if you blow up this section right here, you can see some of these branches are just blown up right over here. Here's the color of the tree root. It's kind of an amber orange. And then the root tips have been colonized by the fungal hyphae. And that's why they're whitish. Some really colonized, very thick blanket. And pine roots always bifurcate in the mycorrhizal roots. So this is a different species. It's a brown hyphal species, the Lephra. But you can see that's the mantle. And you're not even seeing the plant in this area. So there's a lot of fungal tissue there. And if there's C14, it's going to light up for sure. Look like a plant, but in fact it's fungal. Okay, so let's continue to build up this idea of the complex network. We have 3 different individuals of plant and 1 individual of fungus. Now we go out into the real world. This is a stand, probably in Canada, but it might be in near Corvallis, Oregon. Okay, this takes a little bit more explaining. This is a mycorrhizal root tip of a particular species called Rhizopogon. And it looks like a little truffle. It's got a skin, and they're about the size of the tip of your pinky in terms of how big they are. And they're completely colonized by 1 species of fungus, the Rhizopogon, vesiculosis or vinicoid, but that doesn't matter. And then they've got that skin. If you cut them in half, if it's a truffle, you'll see the spore-bearing tissue, and you'll smell it like a mouse would, and you'll say, gee, I think I'll eat that. That's pretty good. But this is not a truffle. These are the root tips. And you can see them. You can see that they've been sliced. That's plant tissue, the brownish color. And then the whitish color in between is the fungal tissue. And then the fungal tissue on the outside, called the peridium, is full of all kinds of nasty chemicals to prevent insects from biting in there. Okay, so that makes a very nice sampling opportunity for Byler. He went out there and he pulled those little root tips up. That's what all the black dots are. So this is a stand of Douglas fir. The green saw blades, I call them, are the trees. Right? So there's a lot of trees, and that gives you a sense of the scale. We're talking, I think, 25 by 25 meters or something along those lines, maybe 50 by 50. A lot of trees, and then they sampled root tips. He sampled root tips. And in a lot of cases, he would sample them, and so he could take it back to lab and identify the individual trees. So you could say that root tip was formed by this tree. Okay, and that's it. But then he could go to other trees and say that tree is associated with a lot of different root tips in this stand. And this was her first mother tree. She said this is the one that's driving the whole system in this particular stand. But you can see there's varying numbers of root tips that were assigned back to a tree. And they're not just with one fungus, they're with multiple individuals of that fungus. So again, you harvest these, and you can have genetic types to identify what plant that came from. Initially, they're harvesting within this box, but then they found one genotype was all over the place, so they had to go outside the box, and then they found her. She's out here. Okay. Now note there's all those little polygons. They draw circles around the black dots that all belong to one individual fungus. one of the rhizopogon individuals. And so once they did that, they could draw, what they call polygons. I call this kind of a bizarre gerrymandering map. So people don't know what I'm talking about, but I think this year they do. And they don't really cross over. They don't fuse, right? The fungus gets its resource space, that is the root tips, and it does not want to give it any of that up to the other fungus. So those black dots are the mycorrhizal roots. The shaded areas are the individuals of the rhizopogon. Incidentally, my wife is on this paper because she did all the genetic typing, or gave them the tools to do the genetic typing of the fungi. They were nice enough to put her on the paper. Okay, so now we have a lot of Douglas fir individuals and a lot of rhizopogon. It's getting really complicated. Think back to the one tree, one fungus microcosms that I was showing you earlier. And Byler, I'm glad he got his PhD on this one because, wow, was that a lot of work. And it wasn't the only place he was doing this. Guy was amazing. And I want to re-emphasize that the fungi are not fusing, they're staying separate. And I'm going to go into that in more detail here in a minute. But for ideas, let's say you put a bag of CO2 that's labeled on this seedling. You hope you sample a tree that's associated with one of the roots that has the fungus that was on this tree also to pick up that signal transferring from mother to seedling or whatever. It's so complicated. I've done so much sampling below. You can't see these things. You can see the roots, but you don't know. The hyphae are invisible to our eyes. So you'd be very lucky to pull that up. And I would say that it would be an amazing, not amazing, and it's happening, but a fairly rare chance that the CO2 sugar is making its way above ground to the seedling over here. There's way too much going on, I think. So she did a lot of lab work to demonstrate that this is happening, but it's always been sort of like, yeah, okay, does it drive the succession pattern in my world? We're not sure yet. We're still working on that. So let me talk a little bit more about hyphal fusions. This is tubervortii, which actually is an edible one for humans. There's a lot of other truffles out there that are too small and too black for humans to harvest and eat, but the squirrels love them, and voles, and whoever else. But what they were doing, Manuela Giovannetti in Italy, in her lab, was looking at how many individuals are forming mycorrhizae on these roots. And in this case, they did a separate experiment where they said, if you grow on a petri dish, you take a single spore and it germinates. This particular one can grow without the trees. That's a rare case for mycorrhizal fungi. But you grow it on a petri dish, and then you take a little, what we call hyphal plug, these little squares. It's agar and the hyphae from a petri dish that had one single spore growing in it. And then they actually took a little bit out, put it onto another petri dish, and grew those up into white lawns. And then they dug out a little bit more and put them back together. Okay, and they grew, and when they came in contact, they fused. The hyphae fused. So that's what this slide shows. They found each other. They're getting closer. They're growing towards each other. They're getting closer. So these are from two different isolates of the same individual. And when they find each other, it turns out they, not always, but often fuse. So by, let's see, this was, what's that? Put your glasses on, Horton. Oh yeah, 25 minutes, 75 minutes, 2 hours, 3.5 hours, 2.5 hours, and almost 3 hours here. And they finally fuse, and you get a continuous cytoplasm. So that's what's happening in here, in this zone, but not so much over in B. They form what's called a barrage zone, and the hyphae recognize non-self tissue, deposit a bunch of nasty chemicals, and run in both directions. They're each doing it. And these are probably not so related, the two spores that were used to generate those hyphal plugs. This one probably with brothers and sisters. We'll call this one the sister, and she's beating up the brother. Stronger. But you can see most of these are not happy to be near each other. They form these barrage zones. This is a classic situation in fungi. So if you've heard of spalted wood, this is a black walnut that was cut near my house at this time. And I was like, whoa, look at that. I've been waiting for that. There it is. And the fungus is probably up Chloe or one of the other saprotrophs. This is not a mycorrhizal story now, but it is a fungal story. And you can see the nasty barrage zone, the nasty chemicals that this fungus lay down. And when it comes in contact with this one and vice versa, they don't cross over that barrage zone. They stay away. They recognize non-self tissue. They want to keep their own resource base. So they don't fuse. They reject each other. And it's very much like we have a similar outcome, but a whole different story is how it happens. If I get a burn on my arm and I need to get a tissue transplant, so skin from someone, one of you, boy, it's just going to get rejected. They can do all sorts of stuff with my immune system, drop it down, and put that. Think of kidney transplants. They got to find a good match for it to work, and for the most part, it don't work. Even if it's a brother, okay, a sister, okay. Twins work best, right? Paternal, maybe. So we are doing the same thing, and I don't think we're doing it to keep competitors out, but probably to avoid parasites that come in with the new tissue, to avoid disease that come in with the new tissue. And this one is a bizarre one that's mushroom specific. You could have selfish nuclei that are in this individual, and as soon as they start growing out and fusing with others, that single nucleus basically takes over the entire system. Don't contemplate that too far. It's a great question for PhD exams. We'll leave it there. So let's move on. So non-self within the same species is they're not fusing. They're recognizing and trying to stay away. Does carbon move easily through that kind of system? I would argue not so much. And then plus there's all those species. So within each of these species, there's ways to prevent things from moving across the membranes from one fungus to the other. And here we have two different species of fungus. They clearly are not happy to be near each other, but it's not so bad, they just ignore each other, right? This one grew, got close, recognized their non-self, and kind of took a sharp turn to the left. To the left, up. And this one is just kind of doing this thing. It's got a fairly thick mycelial mat there. That's going on out there with all those different species. They're not fusing across species borders. Okay. Again, that makes it hard to get sugar to move between each individual. I've also said that between species, within species, between individuals, and then from the fungus, which is heterotrophic, gets its carbon and wants to keep it, over to an autotroph, which can make its own carbon. These are places where there's I have real problems with the idea of carbon moving like that. And if you walk into a forest, this is from a foray. That's what mushroom pickers do. They go on forays and they skip through the forest with baskets. If you ever seen one, ask him which ones he's going to eat or she's going to eat. No, this is from one in Mendocino County. It's a very diverse zone. And you can see just one day, she took this picture from her basket. It was just loaded with diversity. There's a lot going on down there. Even when you don't have the mushrooms, that's what's going on down there. It's that, right? Here's only two, but you can imagine how many individuals here and how do they interact. Most of it is not parasitic, although there are some parasites in that picture too. So let's wrap this up now. Take a deep breath, because this one's going to be really weird. This paper came out in 2003, Scale-Free Networks, in Scientific American, and I said, finally, someone has captured what I think is going on below ground. in these mycorrhizal networks. Totally unrelated to fungi, this paper. This paper was about the internet and nodes of however the internet works, transfer of thoughts and things, I don't know, data. But what you can see is that there's a lot of different colors and they often repeat across the landscape. So this green one is here. Maybe this is the same species over here. Maybe it's over here too. Maybe there's some hyphal connection between these two individuals, but probably not between those two and this one. Pick another color. How about black? There's one there. There's one there. Are they connected? We don't know. There's one down here. They are probably close enough to be connected, within about a meter. We have found that most of the individuals underground are about a meter or less, although there are some that are more like 3 meters, and some, if they're undisturbed, and some that, we had one that was like, this whole stand was one individual Solus at Point Reyes. Again, you have to have genetic tools to identify the individuals. So, you can see how many species of fungi there are, all the different colors. Probably all doing different things for the plants. One's really good at nitrogen acquisition. One's really good at phosphorus. One's really good at rehabilitating after a big fire or quick. Some are getting water, right? Some are just replicates, but I don't think there's a lot of redundancy. In my experience, I bet there's just varying levels of what they do and makes them really important for the plants, and the plants support them and send more sugar to the roots with that one than one over here that is not doing what it needs. Okay, so put your plants on top of that. Imagine me walking into that forest saying, okay, I'm gonna start taking soil cores and separate the roots by the different trees. You better hope there's different morphologies that help with that. Sometimes yes, sometimes no. And then genetically typing the trees, and then genetically typing the fungi on those roots as well. And it's pretty complicated. I don't recommend doing that for your PhD, but if you need help, I can help with the ideas. I'm not going to do this anymore. Okay, so let me just summarize then. I think. Yes. So in my view, the networks are made up of many individuals of many species. Not necessarily communicating as much as we'd like to think. The fungi are patchily distributed below ground, and I would say at a fine scale, a meter or less. Sometimes 3 meters for some species that take a lot of carbon and harvest it and then go out and get nutrients that are really important and scarce, so grow bigger. Oops. Sorry, home people. I really don't believe fungi take directions from the trees. If the tree is delivering carbon down to the roots, I don't think she says, okay, take this to my seedlings, and the fungus goes, yeah, that's a good idea. I don't need sugar. I don't need this. And so, yes, it will get to the seedlings, but it's not under the direction of the tree or even the seed. It's just dispersed throughout the body of the fungus. And we still need better data to know what's moving from fungus to the plants. I think we're still sort of hand waving. But it's a great time to get into the field because someone's going to figure this out better. And they too will get a nature paper out of it. Or a job, as it were. I think the carbon moves through the networks. A lot of people call it through bulk flow. That'd be like, fertilizing and then the water just washes the roots through. I don't think so. I think it's occurring within the hyphae. And in those hyphae, there are cell walls and cell membranes and a lot going on to move material down the hyphal channels. And that's not bulk flow. There's a lot of molecular activity there. So another place that we need more information. We have some good information from lab experiments, but we need to incorporate that into a mother tree model better. And with that, I show you a batik that's in my house from Maasai warriors standing on the shoulders of giants. These are the people that impacted me, my thinking, or were kind enough to listen to my ranting and raving. No, we had great discussions. It's Tom Parker from my master's. Dennis Desjardins and Harry Tiers are two phenomenal taxonomists in mycology, and they were in the same building, and I got a lot of good mycology advice from them, especially Harry. He was, at the time, he had to be close to 80, and just loved having a little neophyte asking him questions. Apparently I asked the right questions, because he was always willing. No, he's just a super nice man. Tom Bruns, my PhD advisor, brilliant. Did all the genetic typing tool development back when PCR was a new thing. And I got into that lab at a perfect time. I saw him a few years ago at a meeting and I said, yeah, I don't think I could get into your lab now, could I? He said, I couldn't get into my lab now. The kid's coming up, my gosh. Okay, Randy Molina and Jim Chappie for my postdoc. A lot of people like you guys at meetings and students in my classes. And then all the funding through the years. So, thank you very much. I'd be glad to do it all over again if you want. Yeah, good. Me neither. Any questions? I try not to ask that question because it's such a loaded question. But I feel it.
I have a question.
Yeah.
So, with the mother tree, are you saying we don't have enough information just to say that like she's providing for all these baby trees and it's just a rich environment for other trees to grow, or?
I think a little of both. She has excess sugar, and she's giving 50% of that sugar is going below ground. What the fungi do with it is not under her control. So it's a resource-rich environment. But I think as soon as she fixes the sugar and it gets into the fungus or seeps into the soil, who knows? We don't have good data on that end. You buy it? This is my, mycocentric curmudgeon viewpoint. The students used to really get pissed at me.
Do I buy it or not? I think there's a lot of complicated things going on that we don't understand. I'm a gardener, and so I'm trying to make sites that are welcoming for plants, but yeah, I don't understand how sometimes it works and it doesn't.
So for I'll get you in just a moment. For my research, what I was showing is how the fungi of one species of tree, and I did it in a lot of places, including around here with chestnut, how the fungi in the stand, if chestnut's been missing, can you put a chestnut seed out there and will it get colonized by the fungi that are on the red oak or the beech or the birch? And the answer is absolutely. They're all, quote, sharing their fungi, which, boy, say that in a PhD lab when you just start. Plants don't share their fungi. What the heck are you talking about? Except she was French, and I won't give you her French accent. Other than to say her name, Monique Gardes, Gardes. And when people called for her, she got tired of it in the lab. And you'd hear her yelling into the phone saying, "My name is not Gardes," because we're in kind of a Latin California area. It is God. Perfect, Monique. Just keep doing it like that. She's great. She did that on purpose. So, yeah, I forgot where I was going, but yeah.
Yeah, I was thinking when you showed us that first slide and you showed the factors that you were trying to rule out, that I was curious whether mycorrhizae are sensitive to pH.
Yeah, thank you. So the question here is, based on the very first slide, where the Douglasaurus is getting established in the Arctostaphylos, and I was trying to figure out how the fungi were involved with that. And she's asked, are the fungi sensitive to pH?
Knowing that Arctostaphylos likes an acid environment.
Yeah, and so they love pH 4 to pH 5.
Douglas fir as well?
Oh yeah, ectomycorrhizal, and it varies to some degree, but not like orders of magnitude up to 7. There are some that get up that high, but they don't like basic. And when you got a basic soil, it's bacterial associations largely. There are some species that have honed in on that niche and do well, and so you will find them there. But by and large in our forest, it's acidic soil.
So we're less apt to find great quantities of mycorrhizae in a high pH environment?
I said bacteria, but also the arbuscular mycorrhizal types. The Glomeromycota likes it slightly basic, 8, 7, maybe down to 6, which is just slightly pH, slightly acidic. But yeah, you're shifting the mycorrhizal type to your Glomeromycota, and bacterial organisms do better in more basic soils like that.
But not generally the prevalence. Like there are more mycorrhizae that are sensitive to a low pH than a high pH.
In that world, the ones that we collect mushrooms from, though, the ectomycorrhizae like the low pH. The arbuscular mycorrhizae like a field or your garden like it higher. And I mean, I could come back. I taught mycorrhizal ecology. I can't believe you never took that class. We could spend a lot of time getting into those dynamics, and you can't really lump all mycorrhizal fungi into one type and they all behave like this. It's kind of what we're doing with the network ideas, though. We're saying, oh, it's a network, boom, we're done. And I'm trying to say, it's complicated. Well, that crazy below ground network image I show, it's really complicated. But throw onto all of this that if you have something like poplar, willow, they form associations with both types. So it starts to get really interesting. They're really good at early succession, where it's more basic, but as the soils develop and become more pH, more acidic, then they are still doing well, but they've just now switched to a different fungus. They don't switch, but a different fungus becomes prominent. Trying to keep the mental stuff out of the plant and fungal world. All right. Oh, wait, yeah.
Any advice for gardeners? Does any of this transfer into gardening or helping grow whatever?
Yeah, so the question is, any advice for gardeners, and does any of this information help with what you're trying to achieve? Who said that? Someone out in the audience said, no. Yes. So when I told you I put Arctostaphylos uber ursi in, or did I tell you that? In my garden. Because I was using it it's a unique root morphology from the mycorrhizal roots. And I would use that as teaching material. And I could never find it when I wanted it. So I planted some in my garden. Well, how did I inoculate those? I went to a spot where there was Arctostaphylos uber ursi that was healthy, not showing any signs of decay, not showing dead leaves, things like that. And I harvested the roots and the soil from that, and then I dispersed that into my garden. So if there's a plant that you're interested in, just go where there's healthy plants of that type and take some of the roots and soil and put that in with yours. And you can really break it up. Post-fire stuff we were doing in California, you could take a thimbleful of soil and put it in with a new seedling, and you get mycorrhizae with that fungus at the new spot. That was an extreme, but it's, a quart would be enough to disperse in a square meter, let's say. Thank you so much.
Thank you, Dr. Horton, very much. We appreciate you sharing your life's work with us, and just thank you very much for demonstrating what really good scientific exploration looks like. It's good to listen to that. And then for our Hamilton students in the audience, obviously declared some gaps in the body of knowledge that need to be filled. So as you think about going out in the world and leading lives of meaning, purpose, and active citizenship, there's an opportunity for you. So I invite you all to join us again next month for our continued Arboretum speaker series, March 7th at 10 a.m. So mark your calendars, where we will join Maggie Riley from our local Wild Ones, Mohawk Valley, for her sequel to a previous talk, The Power of a Compassionate Landscape. So again, March 7th, 10 a.m. Hopefully the environmental conditions will be a little more inviting. Anyway, so again, Dr. Horton, thank you so much, sir.